We have successfully placed human footprints in the lunar dust, but building permanent outposts on other planets requires overcoming a massive engineering hurdle. We can build rockets capable of reaching Mars. The real bottleneck is our own biology. Every cell in our bodies evolved under the constant downward pull of Earth's gravity. The earliest stages of conception rely on that exact physical force to function. Researchers at Adelaide University designed an experiment to test exactly how mammalian reproduction reacts when that one constant force is removed. This diagram illustrates a dual-axis 3D clinostat. By continuously rotating biological samples across multiple axes, the machine randomizes the gravity vector, simulating near-zero G conditions without the deadly interference of deep space radiation. If the physical mechanics of reproduction fail in weightlessness, a multi-planetary settlement is impossible. A colony that cannot reproduce is a temporary outpost. The researchers place sperm from mice, pigs, and humans into microchannel mazes that replicate the female reproductive tract, then put them inside the clinostat. The clinical data showed an unexpected result. In microgravity, the sperm did not lose their swimming speed. Their mechanical motility remained completely intact. This visualization compares sperm trajectories. On Earth, they use gravitaxis, relying on gravity to track along the walls directly toward the egg. Remove gravity, and that signal disappears. Sperm tumble aimlessly, flipping upside down. The result is a 30% drop in successful fertilization. Mammalian reproduction physically breaks down when the mechanical force of gravity is subtracted from the environment. To fix this navigation crisis, the team introduced a chemical intervention. The egg naturally secretes a hormone called progesterone. By flooding the channel with a highly concentrated dose, much higher than the body naturally produces, the chemical beacon overrode the lack of gravity. Human sperm regained their ability to navigate. But reaching the egg is only the first hurdle. The researchers then monitored the zygotes that actually managed to form under these conditions. Exposing early embryos to microgravity for 24 hours caused severe damage. The weightless environment delayed cellular cleavage, reduced the total number of cells, and severely disrupted the cascade of epigenetic changes required to form a healthy blastocyst. Surviving the transit is not enough. The absence of gravity directly threatens the genetic instructions required for early life to divide and grow. The data presents a strange paradox. Microgravity drastically reduces the overall number of successful fertilizations, but the rare embryos that do survive exhibit astonishing traits. Researchers ran an assay on the human sperm that successfully navigated the weightless maze. The surviving sperm showed unusually high-quality markers, and animal blastocysts formed under microgravity showed an increased ratio of inner cell mass and epiblast cells. These specific cell lineages are the raw material for the fetus. Higher concentrations of OCT4 and nanobl markers mean exceptional developmental potential. Space acts as a brutal evolutionary sieve. It strips away the majority of gametes, leaving behind only the most highly functional, robust biology. This elite subpopulation of gametes may possess uniquely resilient mechanosensors, allowing them to sense their environment and adapt even when weightless. To support these resilient cells on long-duration missions, future space settlements will require strict medical interventions. We will need engineered gravity systems, precise timing protocols, and artificial hormonal support to bridge the biological gaps. Mammalian biology struggles heavily without Earth's gravity, yet a fraction of our cells possess an innate, surprising adaptability to the harshest conditions in the universe. The physical toll of leaving our home planet is steep, but the resilience of these surviving cells suggests that a self-sustaining human future among the stars is within our reach.